Hello, everyone. I'll go ahead and start the series of videos covering chapter 5. So I'm going to be dividing this chapter into 5 videos. The first one, this video specifically, we're going to be talking about polymers just to introduce that concept as well as the concept of dehydration synthesis and hydrolysis. So let's see. The reason we want to talk about this is because a lot of the macromolecules we are going to be talking about in this chapter and beyond are polymers. Polymer means many parts. Okay, that poly part of the word right here. Okay, that's many. Every time you hear the word poly, I think we all know that it just implies that it's many. And then we have mer, which in this case, it's kind of just standing for parts. Okay. And each one of those little parts that make up a polymer are called monomer. Very convenient. Mono, it means one, okay? So one part make up many parts once they put them all together. So the analogy that we tend to use to exemplify this is the idea of like a chain where every link represents a monomer, okay? And then the whole chain will be the polymer. Same with a train, okay, where you have many little monomers, which would be each one of these little sections in the train. And being all together, they form a long chain, which in this case would be the polymer. So we're going to be talking about 4 biologically important molecules. 3 of them are polymers. We have carbohydrates. Carbohydrates, we also call them polysaccharides. It's a chain of monosaccharides. And up here, I show an example of it, where basically each one of these little parts over here, okay, it's a monomer. So specifically, this is a molecule of starch. We'll go over starches when we, in the next video, talking about carbohydrates. But each monomer is basically a glucose molecule. So once you put a lot of glucose molecules together, one right after the other, that makes up a polysaccharide. We also have proteins. Proteins are polypeptides, okay? There's that poly word again. And so now we have basically a bunch of amino acids that are chained together, okay? This image over here, it is showing a polypeptide, and we're gonna go over proteins in a different video. But essentially each one of these parts, let me get a little pen here going. Let's see, so each one of these parts, so like this, I'm gonna separate the amino acids for you. So everywhere where I'm separating the amino acids. So here is one amino acid, here's the second and the third and so on and so forth. So it's a chain of them, okay? One right after the other. Then we have nucleic acids, which nucleic acids are also called polynucleotides. It is a chain of nucleotides. And again, we're going to be looking at that in a different video. But basically is this image on the right, wherein here we have, here is one, here is the other, and there's basically 3 polynucleotides in here, or 3 nucleotides. 1, 2, 3. And of course, they get a lot longer than that. But that is what we call these polymers. They're chains of repeating units, okay? Now, each one of these molecules has a different type of unit, and that is what distinguishes them from one and another, and what gives them, of course, different functions. Now, what we're going to be talking about in this particular video is how those long chains are made, okay? The process of making these long chains is called dehydration synthesis, all right? So dehydration, that term, probably rings a bell for you. Whenever you think about somebody being dehydrated, it means that they don't have water, okay? So in this dehydration synthesis, we are losing water. We're using a molecule of water, H2O, okay? So what happens here is that we're gonna have 2 monomers, okay? Remember, monomer means a part, so part 1, part 2, okay? And if we want them to join together to start forming a chain, they're going to have to give up this portion over here, okay? So right here we have an OH, okay? There's also an OH over here. Do you remember what those are? Those are hydroxyl groups, okay? At least in this particular example. It doesn't necessarily have to be a hydroxyl group, by the way. But what matters is that there is an OH and then another H somewhere else because, well, we want them to form H2O, right? 2 hydrogens, 1 oxygen, okay? So if we have these monomers, we want to join them together, they're going to have to give up these atoms that are highlighted in blue. And then the result is that now they are linked. Now we have part 1 and part 2 are linked and they started forming a chain, okay? In the process, a water molecule is released. That is why we call this dehydration. And we are building something, okay? So look at the word synthesis up here. Synthesis. That means you're making something, okay? In this case, we're making a polymer. Now, to break a polymer, you actually do the opposite, okay? So if you have a polymer that you want to break down, for example, when you have these long chains of starches or carbohydrates, and you need to break out glucose out of it to get energy, okay? That's actually one of the processes that happens in the cell. To break each one of these little pieces, you kind of take one piece out at a time, okay? Remember, we have part 1 or monomer 1 and monomer 2. Let's say we want to separate them. So what we're going to do is we're going to use water, okay? We're basically reversing the reaction. We're going to use water and then look over here. We're basically back where we started, where, part 1 or monomer 1 is back to having the oxygen and the hydrogen atoms. And monomer 2 just got its hydrogen back, okay? Now this process is called hydrolysis up here. Hydro, again, that should remind you of water, okay? Sometimes hydro will stand for water, sometimes it will stand for hydrogen alone, okay? In this case, we're just focusing on the water itself. So we're using water and lysis. This is a term we're gonna come across quite a few times, okay? Lysing means breaking. So we're using a water molecule to break something. In this case, we are breaking that polymer apart, okay? So in class, this is an activity that we were going to be doing, but let me show it here. I'm actually gonna have to get out of presentation mode. But this is just to exemplify dehydration synthesis, okay? So what happens during dehydration synthesis is that you're gonna have separate monomers, right? So in this example, I have monomer 1, monomer 2, monomer 3. Notice that each monomer in this hypothetical example, they all have this atom of hydrogen and they also have an OH group here, okay? So let's say that I want to put this monomer and this monomer together, okay? So I can go ahead and basically take this apart. I need to remove these pieces so that I can bring this over here. Now this is going to stay here. This hasn't left, okay? But notice what happened here. We just made a water molecule, right? H2O. Well, we have 2 hydrogens and 1 oxygen, okay? Let's say that I want to keep making this chain longer. Right now, this is considered a polymer, right? We have multiple parts together. Specifically, we have 2 of them, so we could call this a dimer. Di stands for 2. Dimer, okay? But let's say that I want to make this longer, okay? Once again, I want to bring this monomer over here. So I'm going to have to take these things out of the way. And here, I'll just select the whole thing and move it over. And there it is. Now we have another water molecule that was released to put the monomer together, okay? Now, if we wanted to do hydrolysis, okay, I'm just gonna do it in here. So if we wanted to do hydrolysis, let's say that once again, this is a molecule of glycogen, okay? Glycogen would be made up of a bunch of glucoses together. And our bodies constantly need glucose for energy. So let's say that our body says, hey, I need your glucose pieces to fuel the body. It's going to have to then separate those pieces out of the chain. Okay. So our body's going to go ahead and take this piece out. Okay. Now I can't just stay like that. It's actually going to use water to separate them properly. There, we just used a water molecule. Okay. Now the body can use this or something else. But let's say it wants another one. It needs more energy or it needs more of something. It'll once again do the same thing, use the water molecule to separate those monomers, and now they are free to be used for something else, whatever the body needs. I hope this clarifies this process of dehydration, synthesis, and hydrolysis. I will see you guys on the next recording to talk about carbohydrates.